Welcome, everybody, to the final RIEEE webinar of the Spring 2023 series. We are really excited to have this webinar series to provide an opportunity to bring folks together around different topics of shared research interest. And we will be sending out announcements on the fall lineup over the summer. So throughout today's awesome session, which I'm about to introduce, just watch the chat for some links and please use the chat liberally with questions and interactions. And we will have 1 hour of programmed recorded material, but then our speakers have kindly offered to stay on afterwards for a half hour for anybody that can, who would like to have more in-depth kind of Q&A and discussion. Some of the things in the chat will be about next week's workshop, which is alternative protein manufacturing in Western North Carolina, where we will welcome our speakers here. We're so glad they're traveling to us, and we'll be able to get in depth about this. So be on the lookout. So without further ado, I'm just going to let you know. Actually, could you go back? I'm sorry, Adam. I was just going to highlight that the Good Food Institute is going to be talking to us today. So they're going to get into this great presentation on the critical emerging research opportunity that is presented by alternative proteins. And they will talk to you more about what they are in detail, but they're a nonprofit dedicated to the advancement of alternative proteins as a critical solution to climate change and animal welfare. And we're really lucky to have two of their lead scientists on today. And so I will go ahead and tell you who they are a little bit. Matt Hotze has a decade plus of experience in science and engineering, research, and publishing. And most recently, he served for 7 years on the leadership team of the NSF-sponsored Nanotechnology-Enabled Water Treatment Engineering Research Center called NEWT. He began as the Director of Science and Technology at GFI last summer. And in this capacity, he's focused on growing and strengthening GFI's team of exceptional scientists and specialists, and making me jealous because of their retreats and their very cool out-of-office messages about protecting time for deep work. He also loves diving deep into any topic related to the environmental impact on our food systems, and how we could pivot those systems to reduce carbon and water footprints. He holds a bachelor's degree in chemistry from Notre Dame University, a master's degree in environmental engineering from Rice University, where he and I met on our very first day of our master's program, and have been friends ever since, and a doctorate in environmental engineering from Duke University. So we're really glad to hear from Matt today. And we're also glad to hear from Adam Leman, who is just down the road in Chapel Hill, where he's the lead scientist of fermentation at GFI and is focused on the development of biomass fermentation and precision fermentation biotechnology for food proteins. He earned his BS in molecular genetics from the University of Rochester and a PhD from Drexel University College of Medicine in molecular biology. Adam did his postdoctoral studies at Duke University and concentrated on fungal genetics and systems biology. And at the end of that postdoc, he and collaborators established Mimetics, which is a Durham-based startup company. During his time in biotech, he has implemented gene expression analyses in microbial fermentation for beer, biofuels, and biomass. So these tools have helped to better characterize these systems and offer deeper understanding for strain development and process improvement. So he's got plenty of awesome background to help us, and I'm sure a lot of folks on this call will have tons of questions for both of them. I'm going to pass it off now, and we're going to hear from our two speakers for the rest of the time.
Thank you, Christine. Really appreciate it. And apologies to Adam. Something got crossed up with the slides, and his name got misspelled on the slide. So that's not a big deal. But as we're jumping in today, I wanted to start with two facts. We've all heard about all of the disruptions in the food supply that came because of the Ukraine war, and how Ukraine, basically their grain production dropped by about 34 million tons in 2022. And that caused a lot of ripple effects across the economy, including contributing to inflation and other things that we're seeing still play out today. The other fact is that the FAO has projected that the global demand for meat will double in the next 25 years. Next slide, please, Adam. So what that would mean is if you were trying to satisfy the global demand for meat over the next 25 years, you would approximately have to produce 3.6 Ukraines or like Ukraine wars every single year for the next 25 years in order to satisfy that demand, that future demand for meat. And why that is, we'll get into in a minute, but basically there's a huge impact of animal agriculture, and it's way bigger than this Ukraine war impact that we've experienced over the last couple of years. Next slide, please. So the purpose of why we're here today, Christine went into that a little bit. We're going to discuss how we can avoid this slow motion disaster and the leading role that Western North Carolina can play in the global transition to a healthier food future. I will begin today's presentation with an overview of why alternative proteins are the smartest way forward for people and the planet. And then Adam will take the reins and cover the exciting research, white space opportunities, and fermentation. Then he will hand it back to me for a summary of the funding, collaboration, and workforce development opportunities out there and that are relevant to you at App State and others in Western North Carolina. And then we'll take questions at the end with the balance of our time. We'll also be visiting Appalachian State next week, as Christine mentioned. So we would love to follow up with you as as you see fit about some of these topics. Our hope is that as a result of our time together today, or for people watching this recording, that they'll have a deeper understanding of the role alternative proteins will play in our future. Next slide, please. The key research areas to explore and the key funding opportunities to pursue. As you listen to this presentation, write down the ideas that you'd like to follow up and pursue in your research and teaching, and more specifically for our visit next week. But I did promise to explain why why would we need to add 3.5 Ukraine wars worth of food supply to our supply over the next 25 years. So I'm going to get to that in a second. Next slide. So my background is a little bit odd. I came from a water treatment background and worked in a nanotechnology-driven water treatment center for the last 7 years, as Christine mentioned. And I kept thinking to myself, why am I working on problems that are just end of pipe problems, right? So problems that we're just receiving from an upstream source. And one of the things that we do at GFI is we look at what is the root of the issue. And one of the things that we see as the root of the issue is animal agriculture. And we work in several different ways toward an alternative to that version of our food system. Next slide, please. We work in science and technology. So Adam and I, and actually Nathan, who's joining us on the call as well, are part of the science and technology team. And so we work to advance the knowledge, the people, and the funding that's in the science and technology space. We also have a corporate engagement team that engages with all the way from small startup businesses to huge corporations on the alternative protein space, helping them and getting them the resources they need. And then we also have a policy team that is based in DC and advocates for research funding on alternative proteins, and as well as legislation and other things that open the door for these types of activities. And we have actually affiliates in 6 different regions and over 175 staff at this point. Next, please. So back to the Ukraine problem. One of the reasons why we would have to produce so much grain to make up the difference for this meat demand in the next 25 years is that the calories that we have to put into an animal are inherently wasted through the whole process of building the animal, keeping the animal alive, and then harvesting the meat at the end of that process. So we're talking about an 87 to 90%, 97% food waste problem, right? Just because of the nature of the system. And so you can imagine that this has a ton of impact on the environment in terms of nutrient runoff, water use, et cetera. And this is the reason why, if you want that much meat, you would have to produce that much grain, because these animals eat that much grain to stay alive and produce what they produce for us. Next, please. So one example that we like to use is the land example. So only 71% of the land on Earth is habitable. And of that, only 50% of the habitable land is used for agriculture. And on top of that, the other part of the land here is 37% forest, and we really can't afford to lose any more forest. In fact, we're trying to reforest in a lot of different places. So if we take that 50% agriculture land, 77% of that is used for livestock. Because of inefficiencies, and there's like these tiny dotted lines here, but because of inefficiencies, that 77% of land that's used for livestock only delivers 33% of the protein that the agriculture system produces. And so something has to give here in order to feed 10 billion people, the 10 billion people that are coming by 2050. In addition, that 33% that comes from livestock can be very volatile. Next slide. Having everyone on the call, having just lived through the pandemic and then the ongoing effects of COVID-19 and the variants, we know that there's a major problem related to having these concentrated animal facilities and having them, all together breeding various diseases. In fact, the most relevant example recently that wiped out a ton of animals was the African swine fever virus, which can wreak havoc on protein systems from animals. So roughly half of pork production in China was wiped out by this virus, and this virus had reached the Caribbean in 2021. Fortunately, has not reached the United States through basically like extreme measures by the United States to try to keep this out of the country. But again, going back to that 33% of protein coming from animal agriculture, we're not doing a great job of taking risks out of that system, and we view alternative proteins as a way of de-risking that sector as well. Another piece of this, if you go to next slide, is antibiotic resistance. 2/3 of antibiotics produced globally are administered to farmed animals. So we're taking this very valuable resource, which is antibiotics, and we're feeding these to animals. So there's some very aggressive estimates, not aggressive estimates, but very alarming estimates about how many people, especially in developing countries, will be affected by this in the coming decades in terms of these antibiotics no longer being effective and people dying from basic infections because we don't have other antibiotics to turn to. So let's go to the next slide. This slide just backs up the point from the UN, saying that two of the most poignant problems for the next pandemic will be increasing human demand for animal protein, and then unsustainable animal or agricultural intensification. So these two problems are the 1 and 2 of why we believe, or why the UN believes, and we obviously believe by reading their documentation, that the next zoonotic pandemic will come from animal agriculture. Next slide, please. Next, please. Okay, so and then the other thing is that these risks are primarily foisted upon the global South, because those are the people that keep livestock in close proximity to themselves. So this is an equity issue across the globe. But there is an issue basically with like us causing these zoonotic diseases and then having people that are in poor circumstances be exposed to these zoonotic diseases. And so at GFI, we're really thinking to ourselves, like, how can we work upstream of this problem in terms of animal agriculture? And how can we focus on making better protein products, alternative proteins for people who may enjoy animal proteins now, but would gladly switch to something that has less impact on the environment and people? Next slide, please. So our theory of change at GFI is that rather than convince consumers to change their diets or their behaviors, I mean, everybody likes to cook out on Memorial Day, 4th of July, at least in the United States, right? How about provide just a better product, a product that has less impact on the environment, a product that has less impact on humans, and then ultimately a product that could potentially be cheaper and tastier than the original animal-based product. Next slide. So we have 3 main technology platforms, and we'll really be focusing in and honing in on the fermentation platform. And so I'll leave a lot of the discussion about that to Adam, as you know, he's coming up soon in the deck. But the other 2 that we focus on are plant-based and cultivated. And so plant-based meats are what you see oftentimes in the grocery store. So you're talking about products from big companies that are established, and they're biomimicking meat using plants as the building block. And they are meant to taste, look, and cook like conventional meat. Fermentation and precision fermentation, as I said, Adam will go into deeper detail there, but that's one of the technology platforms that we look at. And then the third segment of alternative proteins is called cultivated meat. And basically that's when you take, you generate meat that is identical to meat that came from raising an animal, but you do it in a different way. You just take the cells of the animal and you grow them up into a cultivated version of a burger or a steak or a fish fillet. And then that is the product that is bought and consumed by the consumer. So we look at all 3 of those platforms, but you may be asking, hey, there also is a environmental cost to some of these platforms as well. It's not like completely environmentally neutral to run bioreactors, for example. So next slide. So we've looked at some of these, us or GFI and others have looked at some of these possible technology platforms, and they perform very well in terms of the environmental impacts. For example, plant-based has the potential to reduce greenhouse gas emissions by 90%, use 99% less land, use up to 99% less water. Fermentation, for example, I just picked out one point about this, and I'm sure Adam could answer questions about this, but 90% reduction in carbon footprint compared to beef, for example. And then on the cultivated side, we've done extensive life cycle analyses in partnership with different institutions, and cultivated shows that we'd have 95% less use of land, 78% less use of water, and 93% less pollution, and 92% less greenhouse gases. So if we're able to scale these technologies and get them out into market appropriately, we could see huge gains in terms of the ability to combat climate change, for example. Next slide. So what I want to end with before I hand it off to Adam is that how other significant investments are being made around the globe, and that how we're on the cusp of significant investments, which I'll go into a little bit more detail after Adam details the white spaces in the United States. So the Israeli government just greenlighted a 5-year budget of $20 million for a sustainable protein research center. This is for all the way from fundamental to applied research. The UK government, just within the last 30 days, invested 12 million pounds into what's known as CARMA. cellular agriculture and manufacturing hub. And so they're really focused on that cultivated meat space. And the Israel Research Center is a little bit more broad, but the UK one is more specific. And then in the US, we see several opportunities on the horizon from the federal government. And we also note that there are many big climate funders that are also taking notice of this space. And we'll go into a little bit more detail about that after we head into the white space portion of the presentation. So with that, I'll hand it off to our fermentation expert, Adam Leman.
Thanks, Matt. I'm just going to advance here. So I'd like to talk to you today about the opportunity areas for technological innovation in fermentation. And first off, I want to start out by saying we take a pretty broad definition of fermentation here at GFI. And often when you think of fermentation, you can think of anaerobic conversion of biomaterials to something else using microbes. And often that's taking yeast and making ethanol, or taking lactic acid bacteria on food and making preserved food. But hopefully what I can show you today is that when we take this very much more broad definition, that when you take any microbe, so it could be a bacteria or a yeast or other fungi or even microalgae, and grow them up on some sort of biological feedstock and have them convert food, the food that they eat, into proteins and fats and things like that, that we have an opportunity here to really capture all of that repertoire of microbiological activity to make new foods. So Matt alluded to fermentation being one of these three pillars. I'm going to further subdivide it just to make things a little more complex and talk to you a little bit about the two main parts of what is the alternative protein space in fermentation now. And that first part is looking at biomass fermentation. So biomass fermentation takes usually fungal, but sometimes bacterial or microalgal systems, and grows up a lot of cells very quickly in a particular feedstock. So there are certain kinds of filamentous fungi that you can grow on nutrients like potato starches and things like that. And in just a few hours, get filaments that are chock full of protein from this microbial growth in that substrate. And those filamentous protein fibers, but also things like single cell protein from bacteria, can then be taken, lightly processed, and then texturized, and then made into a variety of foods. So I'll just walk you through a few of those now. So Meaty Foods is a company that uses filamentous fungi to make fibrous, high protein foods that can replicate things like steak. So this is an example here of one of Meaty's foods. Nature's Find is a company that actually does tray fermentation or mat fermentation of a particular fusarium fungi that was actually identified and isolated from hot springs near Yellowstone National Park. And they take it and they make biomass protein that gets converted to things like the protein that is in alternative cream cheeses and alternative breakfast sausages. My Forest uses converted dairy storage tanks and turn them into mycelial protein fermentation systems where they make an alternative bacon product. So you can see here that their mycelial protein mixed with fat does a better job than a lot of the other alternative bacons that you may have seen out there. Corn is sort of the original example of a mycelial protein that has been used in the food system now since 1985 in the UK and now internationally, where they take mycelial protein, grow it up in these gigantic airlift fermenters in the US and the UK, and then turn it into what is a really good substitute for white meats like chicken. On the other hand, there's precision fermentation, which in the past few years has undergone this enormous explosion in sort of the possibilities of things that can happen. So what is precision fermentation? It's the use of a microbe to make a specific food ingredient, where you grow up that microbe in a fermenter and then you induce it to make something that then can be purified away from that fermentation and then isolated as an ingredient. So I have up here on the slide a few examples. So Every is a company out of California that makes egg white proteins, so ovalbumin, and that ovalbumin can go into food products as a drop-in replacement for egg whites. So here's an example of a meringue that's made with the egg white proteins. There's a variety of things that Every has on the market now that includes their ingredient. You may have also seen Perfect Days milk protein. So they make a whey protein that can go into milk or ice cream or yogurt or any of these other dairy derivatives that people are using. Geltor is a little different. They make collagen proteins using bacteria, and these collagen proteins can go sometimes into the cosmetic industry, and that's where they really got started commercially, but now can go into foods, and you can have animal-free foods like gummy bears, where people who don't necessarily want to eat animal products can find these alternatives. But now, hopefully, in the next few years, they can actually make these proteins cheaper and more sustainably than anybody can make in an animal protein. And then finally, Impossible and Triton algae innovations are making heme proteins. So heme proteins are these iron binding proteins that can enhance the flavor and color of foods, especially alternative meats, and then give that food a meaty taste or that iron tasting bite. And then sort of tangentially, but also equally important, Matt brought up cultivated meat. So this is growing animal protein without the animal. So if you grow an animal protein in a bioreactor, they're a little more complicated than microbes. You need to give them growth factors and other sort of protein cues to keep them growing and differentiating as muscle cells or fat cells. So Loris Bio in India is a company that's trying to commercialize and replace all of these animal-derived growth factors from things like fetal bovine serum that people are using in tissue culture, and replacing them with fermentation-derived growth factors. So already we're seeing at the biomass side, but also on the precision side, that there's commercially relevant and available proteins that are made with these approaches. But these production processes are complicated. So often these cells are engineered, and they have to go through multiple screening cycles to get the right kind of expression systems, especially for things like precision fermentation, when you're taking an animal-derived protein gene, optimizing it for incorporation into that microbial genome, and then expressing it. And then you need to design media around that allows for those cells to grow, but it can't be too expensive because these have to be commercially viable products. Go through process development, where you go from that flask that's sitting in an R&D lab or a small 1 liter or 3 liter bioreactor, and then scaled up to 5 liters, 10 liters, then 100, 1,000, and then hopefully up to the economies of scale that we likely need that are in the hundreds of thousands of liters, and then finally be able to produce these products at a higher working volume. But that's only half of the story. So in a lot of traditional fermentations, doing the fermentation, then taking most of that fermentation product is almost all of that process. Then you can take it, maybe for beer, you can move it to a bright tank or clarify it and then bottle it. But for precision fermentation, and to some extent for biomass, much of the effort that needs to be undertaken is in the downstream processing. And we see that when we try to take something like a protein, and if we're taking that protein, we have to take our seed train and grow up a commercially relevant volume of production fermentation. And then you're left with something that may go on the order of 20 grams per liter to 100 grams per liter of protein production within that volume. So all of that then has to be isolated away from the rest of the fermentation. So that's when we're starting to look at up to 50% of the energy and effort inputs into dewatering and concentrating that product, doing pH adjustment and filtration, and then maybe finally doing something like a TFF filtration, where we're allowing specialized membranes that have very small scale, micron scale pores to take and allow that protein to be purified. And then finally, maybe it has to be dried or put into a high concentration slurry so it can be transported to a consumer product good manufacturer. So I just told you how complicated fermentation might be, but why might this work? Well, we think, and we know that it works because fermentation occurs at scales of up to 600,000 liters. And if we talk about something like the biofuels industry, we're talking about 1 million liters or more per fermentation. So this is something that can scale and has scaled before. Fermentation is relatively low cost. It's been used to produce industrial chemicals, feed ingredients, and other high volume, low cost products. Some things like plasticizers, clearly biofuels. And it's familiar when we get away from those things and back to food, to the food systems. There's lots and lots of microbes that are used in the foods that we already eat all over the world. And then finally, not to diminish the work of plant-based or cultivated meat, but microbes allow for really, really fast cycles. I can take a bacteria or I can take a yeast and I can put it on an R&D bench, and I can actually sometimes sort of watch it grow. In the beginning of the day to the end of the day, you can see the progress and the growth that these cells can do. That means you can do a lot of experiments and explore lots of different situations for feedstock modification or genome modification, for example. And then finally, that sort of speed translates to production, where some production cycles for certain kinds of biomass proteins can be on the scale of hours, and precision fermentation usually somewhere along the lines of 100 to 120 hours. And disruptions in fermentation happen quickly. Think back to the 1920s and think back to your imagination for what was happening there. This isn't an era of biotechnology, and this isn't an era of huge industrial fermentations, and yet it was. So when folks figured out that you could take bacteria to produce citric acid, something like 80% of all of the citric acid market was taken up in just one decade in the 1920s, because bacteria made citric acid so much more cleanly and sustainably than it could be made from extracting it from citrus peels. Now, if we look for a more modern example, we can look to insulin production from the late 70s to the early 1980s. We took yeast and we took that recombinant protein for human insulin and put it in yeast and had yeast express this. And this non-animal insulin that came from yeast was able to be made more cheaply and at higher purity than anything that would have come out of a cow pancreas. There's sort of a similar situation in the food industry in 1990 when the FDA approved recombinant chymotrypsin B, which is the rennet protein responsible for curdling milk during cheese making. And now about 90% of all cheese in the world is made by precision fermentation produced rennet. So precision fermentation enzymes have been in our food systems for a while, and they're certainly a vital medicine for many folks who rely on insulin. Now, I take this from 2020 to 2030. This decade is seeing already an increase in the amount of structural complexity that we can make by precision fermentation. We can make higher concentrations of proteins, which is vital for the economic capacity of us to make these proteins at scale. And then finally, that will lead to lower scarcity of these, where we'll be able to consistently see them on the shelves. So I'll talk briefly about some of the key opportunities in fermentation for research. And I think this is really one of those places where I showed you a lot of commercially relevant examples. However, there's still a long way to go to making it so fermentation-derived ingredients and fermentation-derived proteins can be at the scale and price parity that we need to be at with animal-derived proteins. So some examples of different places where research is highly active now is in target identification and selection, just a matter of looking at all of the different potential proteins we can make by fermentation. Strain development to make those proteins at higher concentration, feedstock optimization, I'll get into that a little bit more, and then manufacturing challenges. And finally, going back to what more we need to know about formulation and product development. So if we take strain development and we think about some of the things that we need to do, one of those is take novel strains and identify new ingredients and new products and be able to make them in ways that aren't as highly controlled as they are now. Some of the strains that we need have a very narrow window of sugar concentration or temperature or pressure or even shear stress that you can't stir them too quickly in a tank. And when we're looking at target selection, there's a connection to human nutrition that we're just exploring now. So I just have two examples of papers that are very recent, one from this year, but one open question was, if somebody has a vegan diet where they're eating mostly mycoprotein as a source of protein, what's the performance and muscle building capacity of these folks? And the journal on the left indicates that they're able to add as much muscle and perform as well in competitive sports. I know I'm going from the bottom here, but the second question is there's a totally different amino acid mix when it comes to fungal proteins versus animal-derived proteins. So what are the health and nutrition differences? And this is actually an active area of research. And then finally, if you look at the nutritional content of many of these biomass and some of these precision ingredients, there's a lot more fiber. So one of the things that we have to ask is what are the consequences of adding more microbial fiber to our diets? And so far, the research has said that more fiber for everyone is not a bad thing. Next, I want to talk a little bit about feedstock and waste valorization. So often when we look at fermentation, we're thinking about, especially in the world of precision fermentation, taking highly purified glucose or sucrose from corn or sugarcane or sugar beets and feeding this to our microorganisms to output a food. Well, microbes didn't evolve on highly purified sugar. They evolved on starches and other potential sources of carbon. So trying to unlock some of those metabolic pathways so that we can valorize things like ag stream wastes or side streams from other food processing, where you could take something like potato starch and feed it to microorganisms to get efficient outputs. And then finally, sort of an even further frontier is using gas fermentation. It turns out lots of microbes are good at taking carbon-containing gases like carbon monoxide or methane, and then actually fixing those into sugar and using them for their metabolism. And then lastly, I want to talk a little bit about our road to high volume and low cost manufacturing capacity, especially here in the US. So if you look at this big graphic here on the top right, we know that there's often really good funding for doing something at the bench in academia and in a research university, and that sometimes you can take that to a bioreactor scale. And then often there's this bridge to manufacturing that needs to happen. That's sort of we call it the valley of death. It's sort of a hard place to traverse, this bridge to manufacturing, before you can get corporate investment. And the nice thing is that some piloting facilities, especially at the institute and university level, have popped up. And these pilot facilities help to demonstrate scale at first, like I said, that 5 liter or 10 liter or 100 liter before these are ready for commercial production. And there are a few great examples of this in the US, like ABPDU at Lawrence Livermore National Labs or University of Illinois and Iowa State's piloting facilities that allow for a really nice public-private partnership and feedback loop between equipment suppliers, feedstock producers, students receiving an education in these processes, the full-time staff that work there, piloting clients who often come back over and over again to these pilots, and then finally the demonstration scale facilities that we're actually starting to see come out of these same facilities and with some of the same talent. So universities have a big role here too in providing some of that early infrastructure that doesn't exist elsewhere, and we're seeing that this is probably the template that we'll have to follow to allow for scale and allow for the technology to keep up, because we'll have to invent new technologies along the way and do new research at the pilot scale. So with that, I will turn it back over to Matt and have him walk you through the rest of the potential of alt proteins.
Thank you, Adam. So I just want to run through a little bit about what we see, especially here in the United States, about the rising potential for this research area, and then cover some of the GFI resources that we have available for you on our website. And maybe you can peruse those before we even visit campus next week. Next slide, Adam. So I wanted to just give a quick example. We actually have our own in-house research grant program. And so that started a few years ago. We provide small research grants in the single hundreds of thousands of dollars range for people to explore research in all 3 of these, the pillars that we've identified in this presentation. And I wanted to call out Amy Roat, who's at UCLA, who was a grantee back in 2019, and now has received $1 million from the NSF in 2022, and that was announced in the latter half of 2022, to continue her funding mainly around cultivated meat and scaffolding that is needed for that cultivated meat. Next slide, please. The other signs that we're seeing, and they're continuing to be positive, is this executive order coming out of the White House. So the executive order was announced last year, I believe, in mid-September, and that was the call to order from the White House that said to the different parts of the executive branch, now it's time to write us a report and tell us how are you going to respond to these challenges that we're facing in terms of biotechnology from other countries, and how will this transform industries in terms of the climate change solutions and the food and agricultural innovations that we've pointed out already in this presentation. And so 5 agencies, including the DOE, NSF, and USDA, co-authored a report, and that report is actually in the Thank you, Grace, for adding that to the chat. And I think it's well worth skimming through. Alternative proteins are mentioned throughout. Next slide, Adam. So in the USDA report, which I'll focus on here, they discuss alternative proteins, including plant-based, fermentation-derived, and cell-cultured products, as we've discussed today. And they also talk about new biotechnologies enabling protein production from waste, as Adam has discussed in his section of the presentation. And it really outlines goals for identifying and conducting feasibility studies for high volume, low cost protein and fat sources that can be used in food. And this does not, I want to stress that this does not obligate the USDA to do or to release research grants in particular different areas, but it really does kind of shape the thinking of these agencies as they're going forward, because they've now collaborated on this and they're thinking about, okay, this is the next wave of things coming out of the Office of Science and Technology and Policy in the White House. So we need to be able to respond to this and actually show them the results from what our research portfolio will look like in the next few years. Next slide, please. So I just want to emphasize that we believe that there's a ripe opportunity to enter this research space, and that pivoting into this R&D space, pivoting into this workforce development space, there's a significant growth opportunity here. Next slide. We've identified several programs that we believe, and these will all be linked from the slideshow. And we are willing to share more information about any of these programs, including any experience our collaborators have had applying for them. And one thing I didn't stress earlier about GFI is we are a nonprofit, but we are also agnostic. We're not advocating for any particular company, any particular university, any particular funder to dominate the space. What we want to see is freely shared information, and we want to see the space to grow as much as we possibly can. I mentioned this a little bit briefly before, but there are larger foundations and funds in the climate space that are potentially making multi-million-dollar investments. At GFI, we are also working to potentially place some of those investments in the near future. And as I said before, we have a lot of in-house resources. Next slide. We've already linked to this, but this is the latest and greatest on the state of the industry for fermentation. If you're interested in this area, I highly recommend that you go through this. It is known throughout the alternative protein space as the definitive resource on the industry of fermentation. And there is so much good data, and I had the privilege of actually reviewing this document before we sent it out. And Adam is a major author, as well as our other fermentation expert, Lucas, put this together with a lot of work and a lot of time, including the corporate engagement team. Next slide. The other thing that we have on our website is a funding database. So if you're curious about funding opportunities, what we try to do is regularly update this, probably on every 30 days or so. We're adding research opportunities to this database. So you can go and check on alternative protein or closely adjacent alternative protein research opportunities on this webpage, which is linked at the bottom of the slide. And I'm sure you'll be able to get over there after the presentation. The other thing I mentioned earlier is we have our own RFP that we release either once or twice a year. This year, it'll be once. This is coming later this summer. The date is still TBD, but if you're interested in applying for research directly with the Good Food Institute, that is coming, and we'll certainly let the team here know when that RFP gets released. We also have resources on teaching and course creation. We have next slide, please, Adam. We have a curriculum repository. So we can, this holds information from others who have developed alternative protein courses, syllabi, slide decks, recorded lectures, and recommended readings. And then finally, I'll highlight our collaborative research directory. That's next. So we have a researcher directory. So if you're interested in partnering with somebody in North Carolina or perhaps in the region. There are others that are working in the space. And so we try to make it as easy as possible to find connections in the space, because we know it's a growing area. We know that it's somewhat still fledgling. So there is an opportunity here for you to kind of map out who's already playing in the space and who you might be able to collaborate with on maybe some larger projects. So I want to say that it's clear that alternative proteins are the smartest way forward for our global food systems. There are so many white spaces available for fermentation research, and the tide is just starting to rise on this entire science and technology space. And we want to work with you to catalyze changes in research, infrastructure, workforce development, and help you capitalize on this opportunity before you. And I'll close with this slide. Alternative proteins have massive decarbonization potential. And you'll see here that in yellow, the Department of Energy in 2017 alone had nearly $6 billion available from the federal government to look at clean energy, renewable energy technologies. And if you look at that little tiny green sliver there, this was as of April of last year. So I haven't updated this slide, but it hasn't changed much in terms of the magnitude of billions to millions. This is just 1 year of the DOE's budget, and you're looking at the all-time investment as of last year from the U.S. federal government in alternative proteins. If the federal government is looking around for the next big climate target in order to reach Paris climate goals, for example, the food and agriculture space is going to be that place. And so we can see that as electric cars are starting to dominate the market and become more ubiquitous, we're going to start to see other sectors looked at very much more carefully. So if you could just imagine that green bar going halfway up the scale, which I think is conservative, there is just so much room here to grow. Besides the interesting technology, besides the interesting white spaces, there's so many signals leading us in this direction. So with that, I want to thank you for your attention and listening to us today and joining this presentation. We're looking forward to coming next week, but I think Adam and I will now take any questions. And actually we have Nathan from our team on the line as well, just in case we have any workforce development type questions or curriculum questions. So thank you.
Thank you both so much. And thank you also, Nathan, for joining. I'm very excited that this is just the beginning of our conversations. And I think you can all see as the App State side why this is such an exciting overlap with areas that the university has already invested heavily in. The obvious one is fermentation sciences as the first standalone program in the nation. We've already kind of gotten way out ahead of this possibility. And then there are a ton of other areas in biology and our strengths in nutrition and supply chain development. And I'm leaving out a bunch, but there are so many different ways that we can look at how this opportunity is already in our lap and how we can be a huge part of it. So next week, as I mentioned, we are going to have a workshop dedicated to this. There is an open to the entire campus session from 1 to 2:30 p.m. on Wednesday, May 3rd, and we'll send out everything that you need to know about that. I think you've probably all received maybe 40 emails from us. I'm sorry, we're just trying to get the word out. So if you have any questions on that, please don't hesitate to reach out. And so that's the end of my plug for next week. I'm going to pass it right now for a couple of questions, maybe 10 minutes in our recorded webinar, just to get a few and comments. And then we'll close that at the hour at 2. And then we will move on to another kind of informal, unrecorded exploration for the next 30 minutes. So Mike Hamburger.
Good afternoon, Matt and Adam, and thank you for that. I mean, like Christine said, this is really quite exciting to be hearing about this field. It seems like there's a lot of overlap with activities on campus already and ways we can contribute. Also, I have a 2 o'clock meeting, so thank you for ending in time that I can talk to you for a minute. My question is really big picture, though. I mean, we could get into the weeds on detail, but early on you showed a slide of efficiency of different animal models for food, and I mean, what it really triggered in my mind is growing alternative protein to feed chickens or to feed iguanas in some ways an easier target? I mean, humans have finicky tastes, whereas feeding our protein producers with poorly made, maybe less appealing protein. I mean, is there a space for that in this world, or is it really it's human food that we're producing and that's the target?
Adam, you want to take that one, or you want me to take a shot at it?
Yeah, I'm happy to. I think from a technical standpoint, I agree with you. There are challenges with what we call the organoleptic properties of say fungal food or even like a bacterial single cell. And a lot of the research for those is around trying to find organisms that have a really neutral taste and can be texturized. And one of the cool things that we didn't really talk about but is available is that in that based protein world, there's a ton of research on new ways to texturize, coagulate, and really structure proteins well, so that you can get something like the Impossible Burger that has that crumble. And there's still challenges. I mean, there's the fat in there. If you're a really sensitive taster, you could taste the coconut oil. That coconut oil doesn't taste like the fat that comes from meat. So there's definitely work that has to be done there. When it comes to feed versus humans, there is this inherent inefficiency here, and I think that's really the big challenge. That even if we go and make feed, let's say feed for especially cows, because the lifespan is long, if you're looking at the time to get to a cow that's able to produce milk, you've already integrated over time for over a year before you can get that animal. And that's really the big challenge, is that once that animal is up and producing, that maybe the caloric conversion is a little bit better in that small time interval, but that time to get there is really high. And same with beef meat. Then that's why those numbers are really high. So maybe on the technological scale, there are things that, and there are efforts to make feed out of fermentation. And we can learn some things from there in terms of new feedstocks. And this is a place where people are a little bit more willing to play with things like using wood waste or gas fermentation. But the hope is that eventually we'll be able to take those efforts and technology and translate them to human food.
Well said, Adam.
Other questions or comments?
So I'll just jump in here. I'm Shea Tuberty. I was one of the initiators of the beer fermentation side of things at the fermentation program years ago. And in the initial years, we were looking for external funding. So we were writing grants to USDA and NC Biotech. And I reached out to the NC Biotech organization for a list of small startup kind of biofarm organizations that were taking waste products and trying to ferment them into something. I guess this falls along your precision fermentation area. And there was a lot of interest in helping App State kind of get a kickstart in what's going on here. And unfortunately, that side of the fermentation program has really kind of been the slowest to get off the ground. I think the North Carolina culture around craft beer just took over for a little while. And that's kind of, not that the craft beer industry is calming down, but at least at the university, I think we see utility in moving in another direction and growing the program there. And I saw Brett on here earlier. He's now the current program director, and I suspect he'll be a major voice next week when we meet Wednesday. And there he is. So maybe Brett, I'm kind of coaxing you into talking about how big our fermenters are. What would we need to grow that space? Sounds like we need another building probably to grow into some of these sizes that we were just told we need to get to industrial productivity size. But seems like obviously a really productive area to go to.
Yeah, I can jump in, Shea. And I mean, I don't think the idea would be for us to have, multiple 1,000-liter vessels. But, I think one very interesting possibility is to have like an incubator space at the Innovation District, possibly down in Hickory as well for workforce development, and have our more research-focused, smaller pilot facilities to kind of guide the industrially scaled, production and fermentation. So it wouldn't be on us, that industrial scale, because that would never make sense for us for that sort of direct revenue generation idea and to scale up to that size. But rather we would be sort of the support for that industrial scaling, of other folks from industry who want to come in and use that incubator space or to support industry through the university and other means. I think that would kind of be the idea. I mean, yeah, our largest capacity right now is 400-liter vessels. We could go a bit bigger than that, certainly, but we're not talking 100,000-liter vessels. I don't ever see that in our future.
Okay. Well, I guess maybe a question for Matt and Nathan then. I'm sorry, Nathan, you don't have a voice at the moment, but what's the need for like a student pipeline for this area? Are there other departments and other food science programs across the country that are already, kind of gearing up for this need?
Well, Nathan can maybe type up something in the chat, but my first response is I didn't have time to necessarily speak about this, but you've opened the door for it now. So we actually have a project that Nathan leads called the Alternative Protein Project, which is a now global project of student-run chapters, and there's a very strong one actually at Virginia Tech that is working on developing curricula and outreach to universities from the undergrad master's level, some PhD students as well, but they're really building the ecosystem from that point of time, because there is absolutely a need for people to be researching in this space, and then learning about how to run these bioreactors as well. So I think in some of the materials that Christine will share, we're talking about how do you reskill. If I think Adam made the joke the other day, it's like, when are we going to reach? Are we reaching the asymptote of like craft breweries, right? And so like all those folks that have been running those reactors, like, well, where can they also reskill into a new and growing industry? And through discussions with Christine and the team, I think y'all might be very well placed to do some of that work as well. It's like a sort of a mid-career reskilling master's degree, perhaps in precision fermentation.
Or certificate.
Adam, would you like to add anything?
Yeah, Brett, I heard you say or a certificate program. I agree. I think there's a ton of overlap when you're looking at a QC lab in a relatively well furnished brewery, or you're looking at the cellar and people operating and doing sampling and looking at things. There's a high degree of overlap. There's actually a high degree of overlap when you look in a bunch of other places too, like the dairy industry and like several other places where somebody, and even fuel handling and petroleum, where people are basically trying to understand how to move a lot of liquid from one place to another and then do that in a way that's guided by the data. So I think there's a huge opportunity as some of these other things go away. Dairy's taken a little bit of a hit from things like oat milk, and petroleum handling has done that. So I think there are a lot of people who have sort of the fundamental skills. And then in fermentation, we do have this history here at App State to think about how people can take those foundations and then transfer them over to the workforce, from operators to QC lab folks and beyond, and back then into research at the university.
Well, this is a perfect chance for us to thank our speakers and come to a close of the formal part of our presentation today. Thanks, everybody.